Hi, I'm Hadley Pinder, and I'll be explaining number 1. So it gives us a normal chromosome and an inversion chromosome, and it says that these two chromosomes replicate prior to meiosis, and it says what is the allele and centromere composition of 4 products of meiosis if there's a crossover between S and T. So here's our 2 chromosomes that they gave us, and we have to write it backwards in order to make sure that our S and T line up so there can be a crossover. So for 1 and 4, they are just the normal chromosomes that we were given. And then for 2, what we'll do is we'll take this section here and it connects to this one right there. So it gives us this with 2 centromeres, and then this one, S, will connect to this big T up there to give us that one.
Yeah. Thanks. Okay, I'm Donovan Wixom, and I'll be working on question 2, which is just the definitions. So, non-disjunction in meiosis 2 produces 50% aneuploid gametes. If it's in meiosis 1, it produces 100% aneuploid. And non-disjunction in mitosis can produce a mosaic phenotype. For part B, meiosis in individuals who suffer a paracentric inversion produce a dicentric chromosome and an acentric chromosome. C, Turner disease is an example of a human disease caused by monosomy, while familial Down syndrome is caused by Robertson translocation. Part D, the human liver can have 4N, 8N, or 16N cells, which is fun. Terminal deletion in a chromosome 5 causes, I still don't know how to pronounce that, chat.
Sounds like cat.
It sounds like a cat. It's really sad. But it produces that syndrome. While an inversion on the X chromosome causes type A hemophilia. So F, a mutation is a change in the chromosome structure that can potentially be beneficial. And for G, when a translocation cross is formed, the alternate segregation pattern can produce viable gametes and offspring. That is 2.
Hey, I'm Christina Cross, and I'm going to be doing number 3. So the first one, part A, how many total chromosomes would be found in the humans that have the following conditions? So the first one is Turner syndrome. That's going to be 45 because it's missing 1 sex chromosome. Then Jacobs syndrome. That's going to be 47 because there's an extra sex chromosome with Y. And Cry-du-Chat syndrome, it's going to be 46, the normal amount, because it's only missing part of the P arm. And chromosome 5, Mueller Down syndrome, is 46. And type A hemophilia is 46 because it's just going to be an inversion. So for part B, which of the following terms is not associated with, Charcot-Marie-Tooth disease, type IA? So it's going to be basically all of them except for diploid. And yeah, so what we got was triploid, aneuploid, reciprocal translocation, trisomy, and non-destruction for the ones that aren't associated. For C, which of the following terms are associated with Klinefelter syndrome? And so that's going to be aneuploid, non-destruction, and trisomy. And then the next one is asking which are associated with type A hemophilia, and that's going to be none of the above because it's just an inversion. And that's problem 3.
Sweet. So I'm going to be doing number 5. just because we have it on the board over here. So number 5 is talking about we're assuming it's a diploid cell in a cat, which contains 38 chromosomes. And so in the following situations, how many total chromosomes would be found in the following types of cats? So part A, we have a triploid cat, which, as we know, is n+1. And so what we do is we divide it by 2 to form the haploid, which is 19. Then we would multiply that by 3, which would give us 57. Part B, the cat is monosomic for chromosome 12. So that means monosomic is n-1, which we would subtract 1 just from the diploid number, so it's 37. Part C, a cat that has suffered a terminal deletion on chromosome 7. We know that doesn't affect the chromosome number, just genes within the chromosome. So that will still be 38. Part D, cat that has suffered a Robertsonian translocation between 17 and 18. So a Robertsonian translocation basically fuses 2 chromosomes together, which would subtract a chromosome from our initial total number, giving us 37. And then part E, a cat that has suffered reciprocal translocation between chromosome 6 and 9, that would just keep the same number, switching genes between a chromosome. So that's 38. So number 5 over here, 4 over here.
Here's number 4. So number 4, we're talking about mice chromosomes. And so it talks about homologous chromosomes, a pair of homologous chromosomes in mice. And so when the chromosomes split, they're going to split into different cells. And so for the first, this is our prophase 1 cell. So when all the chromosomes are together. And so we have our what's the word for it? We have our alternate pair, which our alternate pair is just the pair that are across from each other. So we have this pair and this pair up here, and this pair and this pair. And then this one is our adjacent one, which for adjacent one, we have these 2 and these 2. And then for adjacent 2, we're going to have these 2 chromosomes and these 2 chromosomes. And you can see the crossing over happens between this pair of chromosomes and these chromosomes. Okay, that was it. My name's Brad.